And welcome to Bacteria Part 2. I'm just going to get started. So, last time we talked about how bacteria can be kind of like identified through their shape and structure, kind of the unique characteristics they have that other organisms don't have. And then within bacterial populations, what differentiates one bacterium from another. So today we're going to look a little bit more closely at how scientists classify bacteria. One thing we really want to get out of today is, let's say, a scientist, a microbiologist, wants to identify an unknown bacterium. So they've swabbed a surface, they culture that swab, and they get an array of bacteria. And they want to be able to know, what species do I have? They're going to employ a certain set of tests that tell them what bacteria they are working with. And that's really what we want to get out of today. What are those tests? How can you identify unknown bacteria? So just to go back a little bit. So all living things are characterized into three groups. You have the eukarya, which evolved last. You have the archaea, which are thought to be kind of like, archaea are hard to explain. It's kind of like the the miscellaneous category. They're not eukarya and they're not bacteria. Um, they have the name archaea because they are thought to be old. But really, they're just evolved to the way Earth used to be, and they really haven't changed. They haven't evolved much since. You can see that they kind of branched off here on their own, and then they love the heat, they love high methane content, and they love extreme salt content, which is more similar to how the Earth used to be billions of years ago. And then you have the bacteria. And there's not just one type of bacteria, there's all different types. So we're going to look at how you can tell what species of bacteria you have. So there's this flow chart that you can use to classify bacteria. Of course, first you're going to look at morphology. What shape does it have? It has a spiral shape that really limits the species of bacteria it could possibly be. Rod shaped and sphere shaped, not so much. There's, millions of different types probably. And there's cultural characteristics. How does it grow in culture? What does it look like on an agar plate? There's motility, which we'll take a look at. Gram reaction, which you saw in the Gram stain virtual lab. I'll elaborate on that a little bit. How fast does it grow? What type of respiration does it do? Does it produce spores? How does it react to certain biochemical tests? And then if you really want to get specific, you would take a look at the genetic, sequence. What is its genome? That's getting really specific. And then how does it respond to antibiotics, basically? So this really gets you to the strain level, not even just the species. So first, the motility test. This is a pretty simple test. You're asking, does the bacteria move? So some bacteria move, some don't. If you're able to, determine its shape and its cultural morphology, and you still can't figure out what bacteria it is, another simple test you can do is the motility test. You just take basically a needle and you cover in the unknown bacteria. You stab it into this agar in a tube, and you see if it moves. If it spreads out from where you stabbed the agar, then it's able to move. If it stays where you stabbed the agar, then it doesn't move. And this really just is a test to see, does it have flagella? Is it able to move, or does it not have flagella? Okay, so this is another easy one. Some bacteria move, some don't. So if it does, you've limited the number of bacteria it can be. You're only working with the bacteria that can move. Okay. Another test you can do is gram staining. You're determining, is it gram positive or gram negative. You saw this in your virtual lab. I'll just go over some of the basics. So first step is to apply a stain called crystal violet to the bacteria. This turns the bacteria deep purplish blue. You then remove that stain with oh no, I'm sorry, you don't remove it yet. Then you add iodine. Iodine is a fixing agent. You then decolorize with a very strong alcohol. If the bacteria is gram positive, it will stay that bluish purple. If it is gram negative, this alcohol wash removes that crystal violet stain. And then one last step, you restain, oops, I need to do that. You counter stain with safranin, which is a pinkish red dye, which turns the gram negative bacteria a pinkish red. You need to add this, right? You need to add this counter stain to be able to visualize the bacteria under a microscope, because most bacteria don't have a natural stain. So in order to see them under a microscope, you've got to restain them. But looking at this under a microscope, you'll either see purplish blue, meaning it's gram positive, or pinkish red, meaning it's gram negative. And why do they turn these colors? It's because of that peptidoglycan in their cell walls. So if you remember last time, we learned about this molecule in their cell walls that determines whether they are, or I'm sorry, we learned about this molecule in their cell walls. Gram-positive bacteria have more peptidoglycan. Gram-negative bacteria have less peptidoglycan. So the reason gram-positive bacteria stay purplish blue even after that destaining is because that stain, the crystal violet, soaks into the peptidoglycan and is not washed out by the strong alcohol. Gram negative don't have as much peptidoglycan. It doesn't absorb that crystal violet stain, so when you destain it with the alcohol, it goes back to being clear. And then when you restain, when you counterstain with the safranin, it turns that pinkish red. Okay? Another classification method is to look at the way the bacteria respirate. So bacteria have evolved in certain environments, and in that environment, one style of respiration might be better than another. So, usually in class, I ask you all sorts of questions about each of these, but I'm just going to show you the results. So these would be obligate aerobes. Obligate meaning they have to have oxygen. These bacteria have evolved in oxygen-rich environments and need oxygen. These are obligate anaerobes. These bacteria have evolved in environments with no oxygen, so actually if they're exposed to oxygen, they die. So these bacteria have to stay away from the oxygen. They actually will sink to the bottom of the test tube. Facultative anaerobes are bacteria that prefer oxygen but can use anaerobic respiration when they have to. Although it's not a bacteria, yeast are facultative anaerobes. Yeast are fungus, but they're the same way. They prefer that aerobic respiration. They make more ATP, but they'll use this other method if they have to. Microaerophile means they like a little bit of oxygen. Aerotolerant anaerobes means they are anaerobic. They do not use oxygen, but they don't die if exposed to oxygen. So it's not like this one where they purposely sink to the bottom. You can also classify bacteria based on whether they produce spores. So in times of stress, environmental stress, some bacteria, very limited amount actually, will form an endospore. This is basically a teeny tiny little protein coat filled with DNA. Now, you might think bacteria are already basically little blobs of DNA, but this is really serious. So this is their normal stage. In times of stress, they will enter this cycle to kind of shed all of the unnecessary parts just to become a protein coat and a ball of DNA. This ensures the survival of the species DNA in times of stress. If you find bacteria that produce an endospore, you've really limited the amount of bacteria it could possibly be. So if you're on kind of your last ditch effort, like you have no idea what bacteria you're working with, and then you do the endospore test and it comes positive, you've really limited your pool of possibilities. And then, of course, you can do um, DNA sequencing. So this is getting really, really specific. This is working at the strain level. So this would be wild type, and these would be different mutations of the same species of DNA. This is actually E. coli. So this is wild type E. coli with their Paratrichus flagella, and then there's these different mutations that cause different flagellar arrangements. You can actually sequence. So let's say you find this bacteria and you're like, well, this has all the characteristics of E. coli, but it doesn't have this Paratrichus flagella arrangement. So I don't know if it's if it's E. coli. So you want to do this test just to be sure. You sequence the DNA and you see it matches this mutant. So then you know with with absolute certainty that you have E. coli because you sequenced its DNA. Another thing you can do is see how it responds to antibodies. So antibodies are proteins that attack foreign invaders. So in your body, your immune system produces antibodies to attack viruses and bacteria. So and it's very specific. One antibody is designed to attack just one bacteria. So if you've been exposed to one type of bacterial infection, one strain, you don't have antibodies for different strains. It's strain specific. So I'm trying to think of an example. Strep throat. You can get strep throat multiple times in your life because you're only producing antibodies for one strain at a time. So it's a very specific interaction. You know that if the bacteria you're working with responds or is killed by this antibody, you know exactly what strain of bacteria it is. Okay? So here's some possible results of doing these types of tests. So first, of course, you're going to start out with your morphology. Let's say you're working with a gram-negative rod. If it's a gram-negative rod and it's a facultative anaerobe, it can be any of these. If you're working with gram-negative cocci, your results, your possibilities are really limited. It's only Neisseria. If you have a gram-negative rod, you have a lot more possibilities, okay? And then you'd have to limit it to the type of respiration it does, okay? So you create these flow charts to limit the possibilities of the bacteria you're trying to identify. So here's all of the different bacterial families. Actually, I lied. This is Archaea right here. This is the bacteria starting here and then going all the way around. So I just wanted to show you how many different types of bacteria there are. Yes, they just all look like cell walls full of DNA, but they really are genetically and morphologically unique. You can use these different methods to identify what type of bacteria you're working with. Okay, and then I just want to show you. So this is for E. coli specifically, just to show you what I'm talking about. So E. coli, morphology, cultural characteristics, how it responds to biochemical reactions, which you'll actually take a look at next class, how it responds to antibodies. So you can actually see all of these different tests in action. Okay, so that is it for microbiology, bacteria part 2. I will be seeing you virtually again on Wednesday. Please complete the assignment posted. And I hope you're staying safe. And hopefully we will meet again in person soon. All right. Take care.